All right. Good morning, good afternoon, good evening, everyone, and welcome to our webinar, When AI Meets Biology. So we have been observing a very successful year of OpenAI on large language model. And just yesterday, like several days ago, there was OpenAI Dev Day. There was amazing thing happening on the language model together with imaging and all other stuffs. So, but at the same time, for the last several months, I have had many requests from our webinar audience about Geneformer. So in brief, Geneformer represents a pre-trained deep learning model. In other words, it's foundation model, from which fine-tuning towards a broad range of downstream applications can be pursued to accelerate discovery of key network regulators and candidate therapeutic targets. So today we have great privilege of being joined by Dr. Christina Theodoris. So currently she serves as an investigator at the Gladstone Institute and at the same time hold an appointment as a faculty in the Department of Pediatric at UCSF. So Christina obtained MD and PhD at the same time in developmental and stem cell biology at UCSF, and she completed medical subspecialty training in pediatric and medical genetics at Boston Children's Hospital. And then she went for postdoc fellows in the Department of Data Science at Dana-Farber Cancer Institute and the Broad Institute of MIT and Harvard, of course. And most importantly, she's the main inventor of Geneformer. So please join me to welcome Dr. Christina Theodoris as she shares her work about Geneformer with us today, and we'll follow with a lot of questions. Thanks so much, Christina.
Yeah, no, thank you so much for the kind introduction. All right, sounds good. So yes, excited to talk with you all today about our lab's work on transfer learning to enable predictions in network biology. Our lab leverages machine learning and experimental genomics to develop network correcting therapies for cardiovascular disease. And in my previous graduate work, we developed a framework for network-based therapeutic discovery, where we start with a patient with genetic disease, and we perform human disease modeling, for example, with iPS cells. And using experimental genomics data from these disease models, we can computationally infer a map of the network that's dysregulated in the disease. And having understood this map of what's central, important for the network as opposed to what's peripheral from different regions of the network, we can use that signature to design a network-based small molecule screen, where we screen for molecules effect not just in a single output, but on the network as a whole. And we train a machine learning algorithm to first distinguish healthy versus disease, and then identify which molecules, when treating the disease cells, now cause the cells to classify as healthy. And in doing so, we can identify network correcting therapies that we can then perform in vivo testing with to confirm their efficacy in the 3D organism, with the ultimate goal of bringing these back to the patients that we started with genetic disease to impact their disease course. And so we previously performed this framework applied to calcific aortic valve disease due to Notch1 haploinsufficiency, and identified a candidate therapy that had a broad restorative effect on the network as a whole, as indicated here with the red genes being 100% correction back to wild type. And we found that this molecule was able to generalize to primary cells from patients with sporadic disease, as well as to the in vivo setting, where here the control treated mice that model this disease have extensive valve calcification, as indicated by the Alzheimer in red here, whereas this is the only mouse that had any calcification in response to the molecule treatment, and it was a much smaller area. And so an analog of this molecule is under further investigation towards clinical trial, and we're very excited about this approach of network-based screening to be able to identify molecules that have a broad network correcting effect. But one thing that really struck us through the course of that prior research was that network mapping requires large amounts of data, which impedes discoveries in settings with limited data, like rare diseases and disease affecting clinically inaccessible tissue, like much of cardiovascular disease that affects the heart and the large vessels. However, there's been a rapid expansion in the amount of single cell transcriptomic data that's available from human tissues more broadly. So it's our question whether we could take advantage of all this large-scale data that's now available through the machine learning approach of transfer learning to improve our predictions in settings where we don't have as much information. So this differs from standard approaches where standardly we start with a labeled task-specific training data set, and we perform a supervised training to generate a trained model that can yield predictions on the particular task for which it was trained. But if you'd like to have that model make predictions in a new task, you'd have to retrain it from scratch with new labeled task-specific training data and a new task-specific training objective. So it doesn't take advantage of all the more broadly available large-scale genomics data. However, with the approach of transfer learning, we can start with a general large-scale pre-training corpus and perform a self-supervised pre-training on a generalizable learning objective to gain a pre-trained model that now has a fundamental understanding of the learning domain, which in our case is network dynamics. And we can then democratize this understanding to a vast array of downstream tasks by copying the weights of the pre-trained model to individual models for each fine-tuning task, and fine-tuning with limited task-specific data to gain much better predictions on the task than if we only use this limited data alone without the fundamental knowledge gained from the large-scale pre-training. And in this way, the model is able to transfer this fundamental knowledge to new tasks. So today I'll talk with you about our recent work on transfer learning, leveraging large-scale transcriptomics to enable predictions in network biology, which was published earlier this year in Nature. And so here we took this approach to develop a novel deep learning model called Geneformer, which we pre-trained on a large-scale pre-training corpus, which we call Gene Corpus 30M, which we assembled from publicly available data with 30 million single-cell transcriptomes from a broad range of human tissues. And importantly, this corpus was balanced so that no tissue composed more than 25% of the corpus. And we excluded malignant cells and immortalized cell lines that have a high mutational burden and therefore could lead to misinterpretation of the model for genes, for example, that had gain of function mutations and might be acting very differently in these settings compared to the others. And additionally, we performed scalable filtering for doublets and damaged cells. And so here, for each single cell transcriptome, we presented to the model as a rank value encoding, where we first scale the expression of each gene based on its expression across the entire gene corpus 30M, thereby taking advantage of the many observations we have of that gene's expression to prioritize genes that uniquely distinguish cell state. So for example, ubiquitously highly expressed housekeeping genes would move further down within the rank value encoding and be deprioritized, whereas transcription factors that might be lowly expressed when they are expressed but uniquely distinguish cell state would move further up in the rank value encoding and be prioritized. And additionally, this is then now after having performed that scaling, we then rank the genes based on their scaled value of expression in this particular context of that single cell transcriptome that's being presented to the model. And so this is a non-parametric representation and potentially more robust to batch-dependent artifacts that may affect the exact expression values while the relative ranking of the genes remains more stable. And just wanted to mention this because it was one of the questions that was provided ahead of time, just that transformers are quadratic in time complexity. So in this case, we have an input size of 2048 because that fully encompassed about 93% of the cells in the pre-training corpus. But you know, that was at the time we pre-trained the model 2.5 years ago. And here, we found that this number of genes was a good balance in terms of fully encompassing the cells that we had and also the compute needed to run the model. And so after now performing this rank value encoding, this then proceeds through 6 layers of transformer encoder units, which here are some references for those less familiar with this architecture. But briefly, the more interesting of the components is the self-attention layer, which through the course of the training, the model is able to learn which genes, in this case, pay attention to which other genes within the transcriptome to optimize the learning objective. And it's important that this modeling approach is context aware. So as outputs, you can gain contextual gene and cell embeddings that encode characteristics of the genes and the cells in that particular context, as well as contextual attention weights, so which genes pay attention to which other genes, and contextual predictions, which is especially important for diseases that are affected by multiple cell types, for example. And during the pre-training, we use a masked learning objective, which is a generalizable and self-supervised objective. So we don't require any labeling of the cells, and we can take advantage then of much more data. And here, 15% of the genes are masked within the rank file encoding, and the model is trained to predict which gene is specifically in that masked position based on the context of all the remaining genes, thereby learning through seeing many observations of cell states which genes within the context are informative and impacting the position of other genes. And so over the course of the pre-training, we found that in specific attention heads, Geneformer learned in a self-supervised manner to attend significantly more to genes we know to play an important role in regulating the cells. So for example, transcription factors and central regulatory nodes within a gene network, as well as highly ranked genes in each cell context, which was very exciting to us that the model was able to learn this in a self-supervised manner. And so having gained a fundamental understanding of this network hierarchy, we then tested the model's ability to enable predictions in a diverse array of downstream tasks, including in chromatin dynamics, for example, distinguishing bivalently marked promoters, which are known to indicate developmentally important genes, as well as transcription factor regulatory range, so whether transcription factors are acting in short or long range of their targets, which is important for understanding which targets are most likely to be affected by particular enhancers and regulatory variants. And this all just using transcriptomic data without information about chromatin marks or genomic distance, as well as gene network hierarchy, so distinguishing central versus peripheral genes within a gene network, even with increasingly limited data of only about 800 cells, and as well as transcription factor targets, including in the zero-shot learning context, again, without perturbation data. And finally, in disease modeling, including transcription factor dosage sensitivity, which indicate candidate disease genes, as well as candidate therapeutic targets. And so I'll show just a couple of examples here today. So here we're distinguishing dosage sensitive versus insensitive transcription factors by fine-tuning Geneformer. And we found that Geneformer was able to significantly boost predictions of these candidate disease genes compared to alternative approaches, including SVM, random forest, and logistic regression, either trained on ranks or on counts. And additionally, we found that compared to architectures that are the same as Geneformer or shallower with a retained width to depth aspect ratio, we found that without pre-training, we found that this was not able to sufficiently yield the predictions. So the pre-training itself was really critical to the gain of predictive potential we gained with Geneformer. Here we trained Geneformer to distinguish between bivalently and list-for-only methylated promoters, and we found that here again, Geneformer was able to boost predictions compared to alternative approaches. And here we're only fine-tuning Geneformer using the 56 highly conserved loci of bivalently marked genes that were initially reported in Bernstein et al. In 2006. But then when we tested, now just using that fine-tuned model that had only seen the 56 loci, and now testing its predictions genome-wide, we found that it was able to continue to distinguish between these chromatin marks, again, only using transcriptomic data and generalizing to the genome-wide setting. Here we fine-tuned Geneformer to distinguish genes that were central versus peripheral in the Notch1 network, which is important for calcific aortic valve disease, as well as genes that were activated versus non-targets of Notch1, this transcription factor, and found that again, it was able to boost predictive potential. And here we tested fine-tuning with only 884 endothelial cells without perturbation data, but from a disease state. And we found that Geneformer again was able to distinguish between these central versus peripheral factors with this increasingly limited data. And although we were most focused on gene embeddings and the ability of the model to encode higher order characteristics of genes, we also tested its accuracy in cell classification, in this case compared to neural networks and XGBoost methods, and found that here, when classifying cell types from a variety of different tissues, Geneformer was able to consistently improve predictions, especially in cases that have many classes. So with these increasingly complex multi-class problems, these are the accuracy and macroF1 for the various tissues, as well as some examples of the predictions of the different cell types for some of the more complex tissues. So it was really interesting to us that the pre-training itself seemed to be playing a major role for the gain of predictive potential. And here we tested the effect of the number of cells within the pre-training corpus, as well as the diversity of those cells, either from many tissues or from a single tissue in the non-diverse setting. And we found that larger and more diverse pre-training corpuses consistently boosted the predictive potential in these downstream tasks, which was really exciting for us because, having pre-trained this model 2.5 years ago, we have a lot more cells now than the 30 million we had back then, and we're able to potentially pre-train models now that have an even greater predictive potential. And so overall, Geneformer enabled predictions in a diverse array of downstream tasks with limited task-specific data. And we then designed an in silico perturbation strategy to be able to really probe the ability of the model to encode network hierarchy. And here we have the original cell's rank file encoding compared to the in silico deletion of a single or a combination of genes. And we can then ask Geneformer to interpret the effect that this individual or combination deletions would have on the embedding of the other genes within the context of the single cell transcriptome to indicate network connections, as well as the impact on the cell's embedding as a whole to indicate the effect on cell state. And here we performed in silico perturbation by in silico deleting GATA4, which is a transcription factor that's known to play an important role in cardiomyocytes, specifically in the context of fetal cardiomyocytes in this case. And so here we found that in silico deletion of GATA4 had a significantly more detrimental effect on genes that are known to be direct targets based on ChIP-seq data, as well as compared to indirect targets, as well as housekeeping genes. And here we found that in silico deletion of TBX5, again, using just the pre-trained model in the context of fetal cardiomyocytes, again, for this transcription factor that's important for cardiomyocytes, we found that there is a more significant deleterious effect, specifically on direct targets as previously defined by ChIP-seq, compared to indirect targets and housekeeping genes. And because these two transcription factors are known to act cooperatively within cardiomyocytes, we then tested their in silico deletion in combination. And we found that here, compared to the individual deletion of GATA4 or TBX5, in silico deletion of their combination had a significantly more detrimental effect on these particular co-bound targets compared to what would be expected by the sum of their individual effects, as well as compared to the effect on housekeeping genes, indicating that the model had some understanding of transcription factor cooperativity through the course of the pre-training. Here we tested the effect of this in silico deletion now on the embedding of the cardiomyocytes as a whole. And here, when we in silico deleted genes that are known to play an important role on structural heart disease and cardiomyopathy, we found that had a significantly more detrimental effect on these cardiomyocyte embeddings, and specifically more than, for example, genes that are known to be involved in hyperlipidemia, which is important for cardiac disease, but whose pathology occurs in cell types other than cardiomyocytes, therefore highlighting this context awareness of the model. And so here we were testing the ability to recover these known disease genes, but through the course of this in silico deletion strategy, we also identified novel candidate disease genes. And so we experimentally tested one of the top Geneformer predicted novel transcription factors in cardiomyocytes in collaboration with Ling Shao in Patrick Eleanor Lab and Anand Chopra in the BioBroad Cardiovascular Precision Medicine group. And here in these iPS-derived engineered cardiac microtissues, we found that compared to control treatment, CRISPR-mediated knockout of this top Geneformer predicted target, TEAD4, had a significantly detrimental effect on the ability of these cardiomyocytes to generate contractile force, indicating that Geneformer, through the zero-shot learning strategy, was able to identify a novel transcription factor in cardiomyocytes that we could experimentally validate was truly having a functional effect. And this transcription factor is part of the hyposignaling pathway and definitely warrants additional investigation. So overall, in silico perturbation strategy revealed network connections and candidate disease genes with zero-shot learning. And we then wanted to extend this to an in silico treatment strategy to reveal candidate therapeutic targets. And so here we fine-tuned Geneformer to distinguish cardiomyocytes from non-failing or healthy hearts and distinguish those from cardiomyocytes from hearts that were affected by dilated cardiomyopathy or hypertrophic cardiomyopathy to opposing disease states. And so through the course of the fine-tuning, Geneformer is able to define within the embedding space where these particular states lie. And having done that, you can then perform in silico perturbations, for example, starting with cells from the dilated cardiomyopathy setting and deleting random genes to derive the random distribution, and then identifying genes whose in silico perturbation significantly shifts the embedding of these cardiomyocytes back towards the non-failing state, while also shifting away from hypertrophic cardiomyopathy to avoid potential side effects of this opposing disease state. And so through the course of this in silico treatment strategy, we're able to identify candidate therapeutic targets. And we performed experimental validation of some of these top predicted targets, again, with Ling Xiao and Anant Chopra. And we found that here in this IPS disease model of dilated cardiomyopathy due to truncating mutations in Titin, which is an important disease gene for this condition in humans, we found that the control treated cells, they have a decreased level of the ability to generate contractile force compared to wild type. But when we performed CRISPR-mediated knockout of two of these top Geneformer predicted targets, we found a significant improvement in the ability of the cells to generate contractile force compared to the Titin heterozygous. And we found the third target showed a trend in the positive direction, while the fourth showed no effect. But overall, we were very excited to find that the model was able to distinguish these candidate therapeutic targets. And for us, this was especially exciting as a way that we can utilize the model to make novel discoveries, as well as to computationally prioritize the tests that we are going to do experimentally. Because especially when we're thinking about combinations of genes, the number of different tests that one could do experimentally is astronomical. It's not always feasible to do so or cost effective. And so here we can use computational approaches to be able to prioritize which experiments to do in the lab and to test these few examples to have a higher hit rate and then proceed with those that show efficacy for additional testing and proceeding towards testing with clinical trials in patients. And so overall, Geneformer is a foundational deep learning model that can now be fine-tuned towards a broad range of applications to accelerate discovery of key network regulators and candidate therapeutic targets. And here, I talked about a few examples today, but I wanted to make sure we had a lot of time for questions. I know a lot of people have particular questions they want to talk about. So here I'm just including a kind of more comprehensive list of the applications that we had for zero-shot learning and fine-tuning within the manuscript, which you can see for additional details of this. And one thing that I really want to highlight is that although the model can be used for zero-shot learning, for sure, if you have data that's specific to your particular question of interest, it's definitely recommended to fine-tune the model to get better representations and information to answer your scientific question. And also specifically that hyperparameter optimization is highly recommended because for deep learning models, it can really be optimizing these training hyperparameters like learning rate and schedule, et cetera, can really be the difference between no random predictions, basically no efficacy of the model to AUCs of 0.95. So it's really important to do that for each individual task. And, when we first pre-trained the model 2.5 years ago now, and we, were at that point, had comparisons to other alternative approaches. But now we're really excited about the shift in the field to being able to take advantage of all this large scale data and really generating these foundation models that can improve our prediction in settings where we don't have as much information, as well as to gain kind of more higher order predictions for the types of biologically meaningful questions that we all have. And so I wanted here to include a few important points on benchmarking now that foundation models are a growing approach that would be important to think about when training your own models and comparing between different approaches. So here, I think one of the critical points is that, as a field, I feel that benchmarking on biologically meaningful tasks is definitely very important. So, we took care to benchmark on a diverse panel of meaningful tasks, like predicting disease genes, et cetera. And I think this is something that's really important that we as a field, come to a good panel of tasks that we can use to understand the ability to make predictions that are really promoting our understanding of biology. And specifically, one example like clustering in 2D, it's helpful for visualization, but it doesn't really capture all the multiple contrastive layers of information that this information might encode. And so, you could, for example, pre-train Geneformer with the contrastive learning objective of, distinguishing different types of cells, and it would easily perform incredibly well at clustering them in 2D. But that would really reduce the generalizable information to the other axes of features that are distinguishing the cell states. So wouldn't necessarily, although it performs very well in this, be a better model overall. And so just, important things to think about. And also cell type annotation and batch integration is something that, is very important as initial step to be able to then analyze the data. And there's a lot of really amazing tools out there to do this. And so, these are kind of two aspects that, I think there's already a lot of great tools, but the ultimate goal is to make novel discoveries. And so, my hope is that these foundation models are used to really push our discoveries and understanding into some of these new areas that is not possible without all of this large scale data and fundamental knowledge gained from the pre-training phases. Additionally, I think benchmarking on sufficiently challenging tasks is critical. So, we took care to include a range of difficulties in the tasks where alternative simple approaches had random predictions. And this is especially important, as we gain more and more powerful models with more and more pre-training data. So, if there's already a task that simple approaches can perform well in, there's no need to use one of these models. We really want to be using these to push our boundaries of what we can understand biologically. And as we gain more powerful models, we're going to be able to hopefully make predictions in more challenging tasks. So making sure that the benchmarking has a range of tasks will allow us to fully evaluate the maximum potential of the models. And then additionally, benchmarking should include generative and non-generative tasks. So, for generative models, it's, really exciting to be able to perform this generation. And so we should be also demonstrating the abilities to do that. And encoder-only models like Geneformer are not expected to perform generative tasks. So it's important to have both, I think, to be able to, again, fully understand the abilities of the models. And finally, demonstrating the model's ability to enable novel discoveries is really critical. So, here I showed you that Geneformer was able to discover a novel transcription factor in cardiomyocytes with zero-shot learning, and also novel therapeutic targets with cardiomyopathy by in silico treatment. And we experimentally validated these with contractile force measurements in the cardiac microtissues. So, for us, I think, it's really important to be able to demonstrate the model's ability to make these novel discoveries, and then also testing them to show that they're biologically true, which again will, hopefully increase confidence in the models. Which, of course, we're never going to be going straight from these models to a clinical trial. We're always going to be testing these discoveries, but again, I think it's really important to be able to help computationally prioritize the search space and be able to direct experiments towards the most high yield novel discoveries. And then additionally, there's a lot of different philosophies in terms of using prior knowledge versus learning directly from the data. But prior knowledge may definitely boost predictions in tasks where information is already out there in the literature. Because if we're telling the model something ahead of time, then of course it's going to be able to return that information to us very well. And so again, we want to have these models be able to enable novel discoveries, not just be something that is able to summarize and be something we can query information that's already out there, but really pushing our knowledge. So this is especially important for that factor as well. And then finally, I touched on this earlier, but standardized hyperparameter tuning would be a way to ensure an equitable comparison, because hyperparameter tuning really has such a great effect in deep learning models in terms of their predictive potential. And in our work, we actually didn't perform hyperparameter tuning for the benchmarking ones compared to alternative approaches because we wanted a fair comparison. But I think probably a better approach would be to have a standardized hyperparameter tuning where perhaps all the models are tuned for 100 trials with a equitable range in terms of search space, and then be able to identify the best model from that, so that we can ensure we're truly representing the maximum capabilities of the models. And so overall, for us, it's really exciting to see the shift in the field towards foundation models and taking advantage of all this large scale data. We're really excited about all these models and new approaches coming out, and seeing where the field goes with identifying the best ways to integrate this information and being able to push our knowledge in biology. And in the future, our lab is leveraging machine learning and experimental genomics to continue to address these major challenges in network biology. We are expanding on the approaches of transfer learning from large-scale data to include modeling approaches that are also able to take into account additional data modalities, for example, spatial information, to understand not just the in silico perturbation in a single cell, what you expect, but also how that in silico perturbation might affect other cells within a tissue or a microenvironment, to understand cell-cell interactions. We're also very excited to extract some of these aspects of network hierarchy that we found to be encoded in the model, and be able to then use that to explicitly infer the connections between genes and understand causally how genes are interacting with one another. As well as to make context-specific predictions of candidate therapeutic targets, which is especially important for us in settings with limited information, where multiple different cell types are involved in the disease progression. We're then using all these computational insights to drive our design of network-based therapeutic discovery strategies, and to be able to make insights within the mechanisms of variable expressivity and incomplete penetrance, which are outstanding questions in genetics. So how a particular variant that in a particular individual has a very different effect than another individual, even within the same family, what are the factors that are really playing a role in driving that process, as well as the mechanisms of context dependency of key network regulators. So often there's transcription factors that are expressed very broadly, but have a really context-specific effect when mutated or have a decrease in dosage. And so we're very excited in understanding what are the factors that are really determining that, both intrinsically within the regulator as well as extrinsically, and other co-regulators or signaling pathways that might affect its role in different contexts, both in space and different tissues, as well as in time, developmentally, and with aging. And so overall, I'd like to thank all of the lab members, as well as our collaborators and funding sources, and happy to take additional questions, as I really want to make sure that we have time for interactive questions and to expand further on anything that I talked about. Thank you.
Thank you so much, Christina. So let's start with the question sections, okay? So the first question actually coming from Gabrielle. There's a recent published preprint comparing SCGBT and Geneformer in zero-shot settings. The preprint showed that neither SCGBT nor Geneformer can accurately predict gene expression in their evaluation data sets. Do you have any comments on these findings? Hence, is it compulsory to fine-tune the model before performing analysis? Is there any analysis that can be performed by Geneformer at zero-shot setting? And I suspect that the the paper, the manuscript that you are talking about is the one from Kastia Kideska from Oxford, I think, and Alex Liu from Microsoft.
Mm-hmm. Yeah, no, thanks for asking that. And I think, again, like this kind of comes back to some of the things I was just mentioning, but let me see if I can go back a little bit here. So yeah, so first of all, so that paper, yeah, they looked at zero-shot learning and they evaluated on 3 things. One was clustering in 2D, which again, as I mentioned, you can pre-train a model with contrastive learning and get really incredible prediction on clustering cells in 2D, but it wouldn't really help to get a generalizable understanding of all the different axes of information that are really important for cells and the genes. So, I think for that, that's something to just think about. For batch integration is another thing that they tested. And I have some slides actually related to that, so I can show that in a minute. But the last thing was in terms of predicting gene expression. So Geneformer doesn't predict gene expression. So through the mass learning objective, what it's predicting again is what gene is indicated in that particular masked position. So you're really just predicting what's in that masked position. And although that's used for the pre-training objective, that's not really the goal of the model. I mean, we're not using it for imputation. We're using that learning objective to have a generalizable understanding of the particular genes in the cell. It's the same as mass learning objective that's used for training natural language models, for example. You're rarely going to be using BERT for like Mad Libs, just filling in the word that's missing in the sentence. It's more in order to have the model gain a good understanding of generally how these different words pay attention to each other, in this case, genes to pay attention to each other, to be able to have understanding of network dynamics that can then be used for downstream tasks. So here for zero-shot learning, we did demonstrate the model's ability to perform zero-shot learning in a range of tasks. For example, gene context specificity, in silico reprogramming, where we can add OSKM, for example, to fibroblasts and show that they shift towards iPS cell state, in silico differentiation. So adding MyoD to iPS cells and showing that it differentiates or shifts towards the myoblast state. The in silico perturbation of transcription factor targets was performed with zero-shot learning. So what I showed with regard to in silico deletion of TBX5 and GATA4 and having a greater effect specifically on their known direct targets compared to indirect targets, or housekeeping genes, and also their cooperativity, as well as importantly, the novel transcription factor that we discovered in cardiomyocytes and experimentally validated that was also done through zero-shot learning with in silico perturbation and fetal cardiomyocytes. So it can be used for zero-shot learning, and we showed that it's able to make predictions that are biologically true and able to be experimentally validated, which for us is especially exciting because there's been a lot of research over the decades on cardiomyocytes. To be able to predict a novel transcription factor that truly has an effect on their ability to generate contractile force is pretty interesting. But that being said, if you have fine-tuning data, why not fine-tune? I mean, it's going to be better to be able to learn representations of your particular disease setting if you're able to fine-tune the model. The purpose of the model is not to just generate this checkpoint that can then make any prediction. It's to generate a checkpoint that's going to then, with fine-tuning, be way better than if you started from randomly initialized weights. You can now start from weights that are already pre-trained to have general information of network dynamics. So we definitely recommend fine-tuning if you have the data. Although that paper indicated it was resource intensive, most of our fine-tuning is done in 10 minutes. So, I mean, obviously it depends on your resources, but it's not something that's the same as the pre-training. The idea is to have this large-scale pre-training be done, and then with just a small amount of data, be able to fine-tune accordingly. And then related to batch integration, because people might have questions about that too. So in terms of batch integration, I included a couple of slides, so I thought people might have a question about that. So there's 2 ways that we think about for batch integration. One is the gene embeddings, which for us, again, was the main focus of our modeling approach, is to understand the characteristics of genes. So here we are comparing in this case, in the blue line, gene A versus gene B in the same cell. And so these are expected to have low cosine similarity because they're different genes. And here we're looking at gene A versus gene A in a different cell, but of the same cell type in the same batch in orange. Whereas in green, we see again gene A versus gene A in a different cell, the same cell type, but in a different batch. And so here we're testing the effect of these batches, for example, preservation methodology, individual patients, as well as different platforms, in this case, nucleus versus single cell RNA-seq. And we find no effect on the gene embeddings by these batches. And so for us, that's really helpful to know because a lot of our testing is on genes. So we want to make sure there's no batch dependent artifacts. And this is with the pre-trained model without fine tuning. Now when we're looking at the cell embeddings, so this is a little bit different because here you're actually including all of the genes within the cell. And in our case, just a simple approach we take is to average to get the cell embeddings. There's other approaches one can take, and probably, if somebody is really interested in getting better representation of the cell, a better way to do that would be to use like a neural network filtering to be able to, to actually take it, which embeddings are really important for the cells, which embedding dimensions are really important for generating the cell average representation. But anyway, in this case, we're using just averaging. And so here, this is an example of some of the pre-trained embeddings. So the original data separates much more by individual patients as opposed to cell types. But here are the pre-trained Geneformer embeddings separate much better by cell types compared to patients. And then the patients that are separating differently actually are ones with different phenotype. And so the model is seeing them differently because of that as well. And it's another example where here, the original data is much better integrated, but again, you see individual effects from different patients, but with the pre-trained Geneformer embeddings, you see good integration of the patients, and the cell type, again, is the major thing that's, kind of separating these. But, importantly, this is a method for visualization. We also have tools to look at a heat map where you can look at all the gene embeddings. So you can actually look at all of the embedding dimensions as opposed to just this clustering in 2D. Because again, we don't feel that it fully captures the different axes that the cells would contain. So yes, these cell types are different, but there's other axes of information that are not going to be captured here because it's just reducing it to what separates them the most when you do this heat map representation. Mm-hmm. And so here for batch integration by fine tuning, so here you can actually use fine tuning as a method to integrate the batches better than if you just use the pre-trained model. The pre-trained model is seeing many different cells and it's many different platforms. So it, has some effect of batch integration by seeing so many different examples. But when you fine tune it, you can actually tell the model to separate cells based on what you want it to separate rather than another axis that you think is a batch artifact. So here, for example, when looking at IPS differentiation, we have iPS cells, cardiac progenitors, and then different types of cardiomyocytes. And importantly, the different types of cardiomyocytes from this paper, it's unclear whether they're truly different types or whether it's actually just an artifact of the genes that are detected in the nucleus versus a single cell, because there is definitely a different proportion of being detected between the two different platforms. But here, if you only fine tune on the single cell data, and then you make predictions on the single nucleus data, you're able to continue to get differentiation between these different cell types. And then the place where you see the most, and confusion between the labels is in this cardiomyocyte 1 and 2, which again could potentially be in kind of artificial separation. But the number of genes that are being detected in these two different platforms are very different. So when you're compressing to a cell embedding with only, detecting a small number of the genes, it's going to lead to that embedding be separately embedded compared to when we have many genes, because you're just averaging them. So you're going to have, different embedding dimensions just based on that. But when you perform the fine tuning, although the original data is quite separated based on platforms here, even compared to some of these other standard approaches, when you perform the fine tuning, the Geneformer embeddings now on the nucleus data, when only fine tuned on single cell data, are still able to separate to a better degree based on the cell type, and then integrate the platforms, with the main distinction being between these two cardiomyocytes that, again, is a different between the two platforms to begin with. So yeah, hopefully that answers, some of those questions that were, had been coming up, but happy to answer any, more detailed information about that.
Thanks, Christina, for very detailed, analysis of how, of the benchmarks comparisons. Another question from Amir Bayagan. How much does data filtering and batch integration prior to training impact the result? And also very similar questions from Ian McDonald was, can you provide details of how you arrived at like 15% masking rate for training?
Yeah. Okay. So for the first question, so if the question's about our pre-training, we don't do any batch integration ahead of time. The only filtering we did was regarding doublets and potentially damaged cells based on the percentage of mitochondria, and then also the number of genes that might indicate potentially that there's doublets as opposed to individual single cells. But for people's individual data, when they're fine-tuning the model, I kind of went into that in terms of batch integration just now. Fine-tuning will improve the batch integration, but certainly, if you see an effect of the batch and you're just using the pre-trained model as zero-shot learning, it would be fine to perform batch integration ahead of time. I think the fine-tuning itself, also, if you perform batch integration before, it won't cause a problem in the effect of the fine-tuning, because you're still going to be seeing, in your particular setting, the labels that you're putting on the cells. So even if the distributions shifted from what was seen in the pre-training case, the model will be able to recognize that. And then in terms of the mass learning, so there's a supplemental figure in the paper where we showed 15% compared to, two other potential masking percentages, did a little bit better. But that was originally chosen because that's the standard for NLP and what's used for the generalizable learning objective for mass learning for these types of models in general. But you can, change that. We found 15% to do better than the others we tested. But yeah, that can always be tested if needed.
Yes, also another interesting question, actually. Can Geneformer, be applied to real perturbations at scale? How would you suggest approaching that?
Yeah, so that's a great question. I think something that is important to note for Geneformer specifically is that, so because it's an encoder-only model, it's not generating the next and the next and the next effect of the perturbation. So when we have, here, let me actually just go back to that slide because it'll be easier to talk about it like this. So when you have the original cell and then you're comparing, for example, deletion of a particular gene, this is what the model is seeing. So it's basically seeing, if you performed, let's say, siRNA to knock out or to remove a gene, and then you're taking a single cell RNA-seq time point now, right after that gene goes down, but before any of the other downstream consequences, right? Because all the other genes are the same in the cell. So when we do things like auto-SCAM and see that the fibroblast shifts towards iPS cells, what we're looking at is the direction of the shift. So the magnitude might be small because it's, again, like right after you add auto-SCAM, before any of the downstream consequences, take a single RNA-seq time point. So what you're seeing is what the direction of the predicted shift would be. And you see the direction is going to shift towards iPS cells, even though the magnitude is small. Whereas a generative model, ideally would, if it performs well, would be able to generate kind of downstream consequences. So I think, that's something important to think about for the perturbations. When we perform in silico perturbation, that's what we're testing. And, we use that to perform in silico perturbation at very large scales. So one of the students in my lab performed in silico perturbation for every single gene of every cell in the 4 million cells in the fetal cell atlas. So that's, I don't even really know how many perturbations that is, but it's a lot. 4 million times the number of genes in each cell. And so, being able to do that now, in a really large scale, computationally, he's then able to now prioritize which of these in silico perturbations are really key for shifting the cells between different stages or states, and then go back and experimentally test which subset of those are really going to be most likely to be effective. And so I think that's the goal of performing these in silico perturbations at really large scales, and then moving it to a particular more refined experimental test.
Thanks, Christina. Another question from Luca Rossi. How did you scale rank the gene across different cell type by each study or by 30 million cells, and how to avoid tides among those zero expressed genes?
Yeah, so for the first part of the question, it's among the 30 million cells, but importantly, it's a non-zero median value of expression. So we only include the cells that express the gene. And we did that because we didn't want the normalization factor to be affected by how many of that type of cell, and therefore that type of gene, being present, was in the pre-training corpus. So we wanted to make sure that was balanced. So that's why we did the non-zero median value of expression. Regarding the second part of the question, so actually this is a really important point. For these rank value encodings, first of all, there's no ties, and we have the reviewer kind of conversations like publicly, and you can see the discussions related to that, but we tested and there was no ties. The reason is because you have this normalization factor per gene, and so that scales the numbers, so it's not just counts, so because counts could have ties, right? But when you have that scaling, it's very unlikely that the gene's going to have the same scaling factor and the same number of counts for that particular cell, and then the same ranking compared to the other genes that are present there. And additionally, any gene that is not detected in the cell is not in this rank value encoding. It's removed. It's not present. So you're not getting ties for genes that are zero. This is really important because otherwise, first of all the genes would be tied that are zero, and how would you randomize that? And then also it would be such a waste to perform all this computation on zeros. It'd be like if you had an NLP model and then you had a sentence, and at the end of every sentence you included every word in the English dictionary and said it's not there. The model doesn't need that. The model can actually learn by the absence of those words, let's say. So a movie review, if it has the absence of negative words, it's going to be perceived to be more positive, right? And so similarly, the model will learn from the absence of genes in a cell, information about its cell state. So we don't have any of the genes that are zero. And that also reduces the amount of space that the data takes, and it makes the model training more efficient.
Right, and for the transformer applied on the text, they have this sequence, they have the order of the words. And is this ordering the genes, like trying to make it like having the order to be, fit into the model?
Yeah, that's also a great question. So, there's really been a lot of effort to kind of use a lot of different data sources with transformers, and there's really been kind of an effort of representing these things as sequences to be able to be used with transformers. But attention itself is actually not sequence dependent. I mean, you're learning, which genes are paying attention with their genes, or, words, or whatever it may be. And it doesn't actually have an inherent sequence component to it. So I think for us, something that was really exciting was that through this rank value encoding, we showed that you can actually use the model with any type of data that can be represented by ranks and doesn't have to have inherent positional sequence. So for me, I think of it as like, for example, if you had kids in a classroom, and then you had on the board the names of the kids ordered by how well they did on the final exam, that would give you some information about the different kids and the classroom as a whole. But it would be different than the location of where those kids are sitting in the classroom, right? Like that's a different type of positional information, but here we're using ranks as information for the model of how to relate the effect of these different genes.
Yes. And, text people in many languages, like English, people read from left to right. And what happened if we feed, like, I suspect that the models like take the highest rank first and then process later on. But what happened if we just swap the order and, clearly it contains the same information, but does the model behave the same?
Yeah, that's a great question. We haven't tried doing that. I think, my inclination would be to say that it would be robust to that as long as it was consistent. because it'd be kind of like, NLP models that learn from different languages, where the important part of the sentence might be, like you said, in different parts of the sentence. So I don't think that it's necessarily inherently needed that the top predictive gene be the top in the rank value encoding, but we haven't tested that. So yeah, that'd be interesting to look at.
Right. Another question is from Expo. How does Geneformer handle different type of data? Like, do you plan to have any, incorporate any different type of data or multi-omics into this?
Yeah, we would definitely love to in the future, and something that we're working on for different types of data sources. I think, where there are increasing number of data sets out there where there's a single cell with both transcriptomic and chromatomic stability from the same single cell, that to us would be very exciting. We found through a previous model that I co-developed in my postdoc called Mira that, really having it in the same single cell makes a huge difference, because they don't always occur, in a synchronized manner. And if you don't have them in the same cell, then you're necessarily only matching things that look the same. But if you have the chromatin stability that's, changing at a different time scale, or there's genes that the chromatin stability remains the same, but the transcription is changing. And so, you wouldn't get that information if you didn't have them in the same single cell. So we're very excited now there's more data of the same single cell. But that being said, yes, certainly you can include additional languages, the way that you train models with different languages, or additional modalities, the way that, some models are trained with text, and then, might have a separate model trained by images, et cetera, but can put those together to make a more general result. So there's definitely a lot of exciting ways to do that. And I'm looking forward to more models coming out that look like that in the future too.
Thanks so much, Christina. Another question from Kuniyo Sueyoshi. So I'm currently working on single cell data for cancer. As Geneformer exclude malignant cells in the training, should we refrain from applying Geneformer to cancer cells and limit its use in stromal and immune cells analysis only?
Yeah, that's a great question. So we excluded them because you didn't have accompanying genome sequencing data. So again, if you have a gain of function mutation, the gene might look really different than if you had, the gene in a different cell that didn't have that mutation, and the model would have no information about that. But that being said, you can still use it for cancer. We're having a collaboration right now with the Department of Energy, and they're using it for some cancer applications, and also a collaboration with some other labs that are doing that. I think one important point is, like, when you fine tune the model, you're going to learn those cancer dependent representations of the network. Or you can also extend the pre-training using a cancer data set, which we've also done in the past. The model, even with different types of cells or tissues that was not present in the pre-training corpus, when you fine-tune it, it's going to see that and learn about that representation. So I think it's totally fine to include it. Just know that perhaps if you perform just zero-shot learning, then the model might be giving you the prediction of a gene based on its interpretation of a gene and with its normal state, and not that gain of function mutation. So, and showing a data that's relevant would be helpful to it.
Yes, and there's one last question from Shichen Wu. Really impressive work. Thank Christina. The recorded video will be shared after the meeting or not? It will be shared. The question to Christina is, which licenses apply for Geneformer?
That's a good question. It's on the repository. I don't remember off the top of my head, but it's free and open source, and the license is on the repository, so you can look at it there.
Yes. So the license I check is Apache 2.0, which is
okay. Thanks. I couldn't remember because we were thinking of a different two. I didn't want to say the wrong thing here, but yeah, it is there.
All right. So we have a lot of questions. There are 32 questions and 10 more questions, which is totally about 42 questions left unanswered, but we have limited time. So we will communicate with Christina and answer the questions for you. But again, Christina, thank you so much for having a great talk today and answered a lot of questions, important questions, and also difficult questions, which makes our brains very triggering, and it's really a great talk.
Yeah, no, thank you for all the amazing questions. And yeah, we're really, again, excited about the shift in the field towards using all this large scale data to train foundation models that we hope will gain some really important novel insights in biology. We're happy to talk more with anyone that has questions and support your use of the model. So thank you.